Hello, welcome to Biology, the scientific study of life. Amoebozoans have lobe-shaped pseudopodia. Many species of amoebas that have lobe-shaped rather than thread-like. Pseudopodia belong to the clade Amoebozoa, or Amoebozoans include gymnamoebas, entamoebas, and slim moles. Gymnamoebas constitute a large and varied group of amoebozoids. These unicellular protists are ubiquitous in soil as well as freshwater and marine environments. Most are heterotrophs that actively seek and consume bacteria and other protists. Some gymnamoebas also feed on detritus, which means non-living organic matter. Anatomebas amoebas, whereas most amoebozoans are free-living. Those that belong to the genus Entamoeba are parasites that infect all classes of vertebrates as well as some invertebrates. Humans host at least 6 species of Entamoebas, but only one, E. histolytica, is known to be pathogenic. E. histolytica causes amoebic dysentery and spread via contaminated drinking water, food, or eating utensils, responsible for up to 100,000 deaths worldwide every year. The disease is the third leading causes of death due to parasites after malaria and schistosomiasis. Notice this figure. Slim moles or mycetozoans, from the Latin meaning fungus. Animals were once thought to be fungi because, like fungi, they produce fruiting bodies that aid in spore dispersal. However, the resemblance between mycetozoans and fungi appears to be another example of evolutionary convergence. Molecular systematics places slim moles in the clade Amoebozoa and suggests that they descended from unicellular gymnamoebas. Like ancestors, slim moles have diverged into two main branches: plasmodial slim moles and cellular slim moles, which are distinguished in part by their distinctive life cycles. Notice that. Plasmodial slim moles. Many species of plasmodial slim moles are brightly pigmented, usually yellow or orange. At one stage in their life cycle, they form a mass called a plasmodium, which may grow to a diameter of many centimeters. Don't confuse slim moles plasmodium with the Apicomplexans parasite plasmodium. Despite its size, the plasmodium is not multicellular. It is a single mass of a cytoplasm that undivided by membranes and that contains many diploid nuclei. This supercell is the product of mitotic and nuclear divisions that are not followed by cytokinesis. The division of cytoplasm in most species, the mitotic divisions, are synchronous, meaning that thousands of nuclei go through each phase of mitosis at the same time. Because of this characteristic, plasmodium slim moles have been used to study the molecular details of the cell cycle. Within the fine channels of the plasmodium, cytoplasm streams first one way, then the other in pulsing flows that are beautiful to watch through a microscope. This cytoplasmic streaming apparently helps distribute nutrients and oxygen. The plasmodium extends pseudopodia through moist soil, leaf, mulch, or rotting logs, engulfing food particles by phagocytosis as it grows. If the habitat begins to dry up or others is no food left, the plasmodium stops growing and differentiates into a stage of the life cycle that produces fruiting bodies which function in sexual reproduction. In most plasmodial stem moles, the diploid condition is the predominant part of the life cycle. Cellular stem moles pose a semantic question about what it means to be an individual organism. The feeding stage of the life cycle consists of solitary cells that function individually, but when food is depleted, the cells form an aggregate that functions as a unit. Although this mass of cells superficially resembles a plasmodial stem mole, the cells remain separated by their membranes. Cellular stem moles also differ from plasmodial stem moles in being haploid organisms. Only the zygote is diploid, and in having fruiting bodies that function is in asexual rather than sexual reproduction. Furthermore, cellular stem moles have no flagellated stages. Notice that the life cycle of a plasmodial stem mole. Notice the meiosis. Notice also the germinating spores, mature spore. Notice also mature plasmodium preparing to fruit and feeding plasmodia and snigmy. Notice also Dictyostelium, discoidium. Dictyostelium discoidium, a common cellular stem mold on forest floors, has become a model organism for addressing the evolution of multicellularity. One line of research has focused on the fruiting body stage. During this stage, the cells that form the stalk die as they dry out, while the spore cells at the top survive and have the potential to reproduce. Scientists have found that mutations to a single gene can turn individual dictyostelium cells into cheaters that never become part of the stalk. Since these mutants gain a strong reproductive advantage over non-cheaters or cheaters, it is puzzling that all dictyostelium cells are not cheaters. In 23, scientists at Rice University and the University of Turin, Italy, discovered why most of the cells do not cheat. Cheating mutants lack a protein on their cell surface, and non-cheating cells can recognize this difference. Non-cheaters preferentially aggregate with other non-cheaters, this depriving cheaters of the opportunity to exploit them. Such a recognition system may have been important in the evolution of multicellular eukaryotes such as animals and plants. Notice that the life cycle of Dictyostelium, a cellular stem mole. Notice also closer look. Notice that sexual reproduction is zygote, meiosis, syngony, and so on. Okay.